The 30 trillion cells of the human body must act as a single cohesive unit. This requires continuous coordination, supported by a network of chemical and electrical signals moving between every individual tissue bed. This network relies on 3 core components. The chemical message itself is the ligand. The structure that receives that message is the receptor. And the process of decoding that chemical contact into an actual cellular command is signal transduction. These signals are categorized strictly by the distance they travel. Endocrine signals move through the bloodstream to distant target cells. Paracrine signals target the immediate surrounding area. Autocrine signals act on the exact cell that produced the ligand. The nervous system utilizes a specialized paracrine variant known as synaptic signaling, where neurotransmitters bridge the microscopic gap between neurons. While distance determines how a ligand arrives at a cell, the chemical makeup of the cell membrane dictates how the cell responds to that message. The selectively permeable phospholipid bilayer defines the cell's outer boundary. Because of its hydrophobic core, it prevents most extracellular information from entering the cytoplasm. Lipophilic molecules, such as steroid hormones, can dissolve into these lipids. They pass through the membrane without assistance, moving directly into the interior of the cell. Hydrophilic or lipophobic ligands, including peptide hormones and catecholamines, are physically repelled by the membrane. They are locked in the extracellular fluid. This physical constraint is visible here. The lipophilic structure glides through the bilayer, while the lipophobic molecule strikes the surface and is forced back. Because these ligands cannot physically enter the cell, they require transmembrane receptors to translate their presence into an internal chemical command. The G-protein-coupled receptor, or GPCR, is the primary translator of lipophobic signals. Its structure consists of a single polypeptide chain woven through the membrane 7 times in a series of alpha helices. The schematic identifies 3 functional domains. The extracellular domain binds the ligand, the transmembrane domain anchors the protein, and the intracellular domain interacts with the G protein. In its resting state, the G protein is a heterotrimeric complex. A molecule of GDP is bound to the alpha subunit, keeping the entire protein inactive. When a hormone binds to the exterior site, the entire protein shifts. This conformational change moves through the 7 helices, altering the shape of the intracellular domain. This shift causes the alpha subunit to drop its bound GDP and pick up a molecule of GTP. This high-energy bond causes the alpha subunit to dissociate from the beta and gamma subunits. The GPCR operates as a mechanical switch, translating the arrival of an external chemical into the physical liberation of an internal messenger. The liberated G-alpha-S subunit travels along the inner face of the membrane to activate an effector enzyme called adenylyl cyclase. This activates a catalytic cycle. Adenylyl cyclase rapidly converts ambient ATP into thousands of cyclic AMP or CAMP molecules. The CAMP acts as a second messenger. As shown in the kinases block, it binds to the regulatory subunits of protein kinase A, forcing the release of its active catalytic subunits. Once active, the kinase initiates a phosphorylation cascade. It adds phosphate groups to specific proteins, triggering immediate physiological responses like cellular secretion or changes in membrane permeability. This architecture creates exponential gain. A single ligand at the receptor can produce 10,000 CAMP molecules within seconds, rapidly shifting the cell's metabolic state. A different class of GPCR engages the GQ pathway. In this sequence, the dissociated alpha subunit activates the enzyme phospholipase C. Phospholipase C cleaves the membrane phospholipid PIP2 into 2 distinct second messengers, diacylglycerol and inositol triphosphate. Diacylglycerol, or DAG, is lipid-soluble and remains in the membrane. It travels laterally to activate protein kinase C, which phosphorylates a specific set of target proteins. Inositol triphosphate, or IP3, is water-soluble. It diffuses into the cytosol and binds to the endoplasmic reticulum, triggering a sudden release of stored calcium ions. By rotating the type of G protein attached to the receptor, the cell uses a standardized GPCR structure to execute entirely different biochemical programs. Enzyme-linked receptors bypass G proteins. Their intracellular domains are built as catalytic enzymes that activate directly once a ligand binds to the exterior. The insulin receptor is a receptor tyrosine kinase. Binding pulls its subunits together, causing the beta subunits to autophosphorylate. This activates the insulin receptor substrate, changing the activity of metabolic enzymes. The leptin receptor follows a different logic. It uses bound JAK2 enzymes to phosphorylate STAT proteins, which then move to the nucleus to act as transcription factors. These systems contrast with the lipophilic steroid hormones, which move through the membrane and require no surface receptors. As shown here, steroids bind to receptors in the cytoplasm or nucleus. The resulting complex binds to hormone response elements on the DNA, directly altering mRNA transcription. Receptor location determines the speed of the response. Membrane receptors trigger enzymatic changes in milliseconds, while intracellular receptors dictate genomic changes over hours or days. When a single cellular signaling cascade is compromised, the resulting physiological effects are immediately apparent and systemically catastrophic. The cholera toxin targets the GS-alpha subunit in the intestine. It chemically modifies the protein to prevent it from hydrolyzing GTP back into GDP. This disables the molecular off switch. The G protein remains locked in an active state, causing adenylyl cyclase to churn out an endless flood of CAMP. This runaway signal causes chloride channels to stay open. The resulting electrolyte imbalance forces massive amounts of water into the intestinal lumen. This clinical collapse stems from the targeted sabotage of a single molecular inhibitor. In healthy cells, every signal must have a mechanism for rapid termination to prevent overstimulation. The system uses a two-part termination. The G-alpha subunit converts its own GTP to GDP, while the enzyme phosphodiesterase actively breaks down any remaining CAMP in the cytosol. Mastering these pathways allows for the categorization of hundreds of different hormones into predictable groups. The flat champ mnemonic identifies the anterior pituitary and systemic hormones that exclusively utilize the CAMP pathway we traced in Act 2. This list identifies the hormones that default to the phospholipase C pathway, notably the hypothalamic releasing hormones and potent vascular constrictors. Understanding these mechanical pathways provides the structural logic for endocrinology, allowing a clinician to predict the behavior of a hormone based on the receptor class it engages.